Thank you very much for that introduction there. Yeah, so uh, essentially, long story short is that I study lakes. So people ask, what do I do? It's like, I'm a biologist. I said, well, what do you do? I study lakes. And then first thing they ask is, so how are the fish? And they're like, I'm sorry, I'm not a fisheries biologist. I don't know much about fish. And then I know, essentially, you can think of it as fish food and below. So all the stuff you really can't see or need a microscope to see. So when I look at a lake and I look at Christine, I can't see Christina Lake anymore, but when you look at the lake, you can see it differently. I see it differently than a lot of other people. I think about all the changes and I think about the lake as a whole and this mixing and different things that are going on. And a lot of people think, oh, I can fish here, or I can swim here, or the lake is clear, or the lake is green. So there's a lot of different things there, but I do look at it differently because it's my life and it's my passion and I love studying lakes. So that's essentially what a water quality monitoring limnologist is, somebody who studies lakes. And I'm with Ministry of Environment. And last time I was here was 2019. And as Grace had mentioned, I've been coming here since 2008, talking about water quality in the lake. So from 2019 until present, we've expanded a regional project. So essentially, it kind of started out as lakes in the Okanagan, Upper Shuswap, and Christina Lake as a regional monitoring program. And over time, it's expanded to the province and covers most of the province. But there are some noticeable gaps. And so we're here at that little red dot in the bottom. But as you can see, that pretty much north of the province doesn't have too much. And that's primarily because we don't have staff north of Prince George to actually get up there. So we've got staff in Smithers, kind of doing up some of the Skeena, and the plan is eventually to kind of cover all of the what are called BEC zones, so the biogeoclimatic ecosystem classification zones. So those are the different ecological zones around the province, and we've got most of them covered. And the hardest ones to get are the ones that are north and up. So we're doing higher elevation and more northerly sites in the future, but continuing the lakes that we have down south, and especially some of the lakes where we've got long-term data sets like Christina Lake. So goals here, there are 5 of them. You can just kind of get through. So essentially, we're looking at current water quality conditions. What are things like now? And then how are things changing over time? And then how essentially human activity, so how are we screwing things up? And then understanding the impacts of human influences. How are lakes responding to climate change? So that's a huge big black box that we're still trying to understand. And you can't just categorize climate change on lakes around the province. They're all very different and they're all very unique. So that's what we're trying to understand is how things are changing in response to these different impacts. And then 5 is kind of what we're doing here is informing. So informing a lot of different people, and one of the groups that we're here to inform are the stewards, the people who live here at the sites. I don't live here. I love Christina Lake, but I live in Penticton. But this is what we're here together is to understand this lake, and I'm here to communicate some of the results that we have of our monitoring program. So in my previous role, I was an environmental impact assessment biologist, and that was really looking at essentially all of the effects of forestry, of municipal effluents, of mining activity, of anything that had an impact to water. We were looking at the effects of that on the aquatic environment. And we have what are called water quality objectives. So while no longer my focus on this provincial scale one, it's still important for folks like you to have these kind of like these benchmark levels to try and understand, well, where should we be? What are we aiming to be? Like above this line, below this line, what are you trying to do? So those, I will have a few graphs here that'll have those water quality objective lines on there. So overall, things are quite good at Christina Lake. So oligotrophic means low in nutrients. And I say very good water quality, but that's could be like several asterisks beside that. It kind of depends, and things change over time, right? But the goal really is to kind of keep it that way. So, Christina Lake Stewardship Society has done a fantastic job, especially with the education piece and influencing what people do on the landscape is incredibly important. And educating people about invasive species is important. That can totally change the lake riparian areas, which I won't get into because that's a touchy topic apparently more recently, but it is wildly important to have these things protected because everything on the landscape has an influence on water quality. So everything is connected. So we've had people talking about forests and wildlife and fish and all sorts of other goodies, but overall, everything that's done on the landscape affects water quality overall. So we've got a couple of monitoring sites, one in the North Basin and one in the South Basin. So a deep site and a relatively shallow site. 26 meters is still deep, but still relatively shallow in comparison. And one has been going since the early 70s and one since the early 90s. And the stewardship group does sampling approximately bi-weekly. It's changed over time a little bit, but they're a vertical profile. So it's looking at from the top to near bottom, looking at temperature and more recently dissolved oxygen. And Secchi depth is a measure of water clarity and how it changes over time. And ENV, measure environment. So we had a way of sampling water quality that was similar to what we do now, but now we've standardized things for all the lakes that I had shown on that first graph, all of the little blue dots. So it's provincially significant, standardized. So this is our standardization here. So we do sampling 2 times per year, once in the late winter, spring, and I say that because the problems are quite variable. So spring on Vancouver Island is in February apparently, but here it is not, and up north it is not. So spring sampling, or it's either just called mixed. So when the whole water column is fully mixed, and then late summer and fall when it's stratified into kind of warmer layers and the colder layers on the bottom. And we do different depths. So the shallow lakes that are arbitrarily less than 10 meters, we do a near top and a near bottom sample for water quality sampling. And then the deeper lakes, we do a composite sampling because for a 50-meter site on the north end of Christina Lake, there's one little scoop of water. There's one little scoop of water at the surface or at the bottom. Does that represent the water quality there? So instead of one, we get multiple samples and kind of pool them together. And then a whole bunch of parameters. So we have physical, biological, and chemical. And biological, again, is below fish. So we do zooplankton, which is essentially fish food, algae, the phytoplankton, which is food for the zooplankton, and then the water chemistry. So because I haven't been here in a while, I'll do just a brief little review about water quality sampling. So this is all this Christina Lake. And we've got the epilimnion and hypolimnion. So those are the layers of water that are separated by the thermocline. So as the lake warms up, you can throw on the next one. So the surface water there on that graph, the top layer of water is warm. So when you're swimming, you'll notice it's quite warm. But if you can get down deep enough, you will reach that cold layer. And those of you who fish will know that most fish will be below that level. They prefer cooler water as opposed to the warmer layer of water. And these are really important for limnology. They're distinct layers that generally don't mix too much throughout the year, and they're distinct units. So that's why we sample both. So from that, we've got our little sampler there. Next. And we take a composite of 3 samples to try and get as representative as we can from the upper layer, and next from the bottom layer as well. So we pull those together, send those into the lab, and get results back. The other thing I want to mention is some people might have heard the term lake turnover. So this is about lake mixing, and the lake doesn't just like turn over and flip. Like it's a much more gradual process of this mixing, but essentially in the summer, as I mentioned here, you get those layers of water. And then in the fall, as the air temperatures cooling down, the upper layers of water will cool down as well, and it slowly kind of gets the water from the bottom can kind of mix up with the top layer. So it's kind of like, it's called isothermal. So the similar temperature from top to bottom. And then ice cover happens, and then it will be, you will get these distinct layers forming under the ice. But as you'll notice, you can see there, like it's 0 degrees at the top and it's 4 degrees at the bottom. So it's a very small layers of water there, but water is most dense at 4 degrees. So that's why it's warmer at the bottom compared to the top. And then as ice melts, you get this mixing event again. And then that's when we're sampling. So essentially spring and summer is when we're sampling. So you get that kind of mixed period and those distinct layer periods. Okay, so what are the key things we're going to be describing? I won't tell you about everything. Like we sample a whole variety of different parameters, metals, ions, all sorts, but I'll kind of focus on just some of these key ones here. So from the water chemistry, total phosphorus and total nitrogen, TP and TN, are key nutrients. Algae productivity, so chlorophyll A, so all plants have a chlorophyll A, and it's that green photosynthetic pigment. So we're measuring chlorophyll A to see how much algae is in the water. So, and these are all linked, right? So you've got water chemistry, so you have an increase in nutrients, you have an increase in algae productivity, and we'll see an increase in chlorophyll A. And Secchi depth and water clarity, if you have more particulates in the water, more algae, more turbidity, then you'll have a worse water clarity and a shallower Secchi depth. And then we can look at vertical profiles for looking at temperature and dissolved oxygen for the entire water column. Okay, so that's my linology primer. So total phosphorus, it's a limiting nutrient for Christina Lake as it is for most freshwater lakes in the province. And by limiting, I mean that it limits growth of algae. So it's a key important nutrient to really look at and focus here. So there are trends that are influenced by climate cycles. So essentially in years of increased runoff, you're going to have increased levels of total phosphorus. So from the landscape, you get more water coming into the lake and it's bringing in all those additional nutrients. And in years when it's drier, then you'll have less of that, generally speaking. So the two graphs here, so we've got north basin on the top, south basin on the bottom, and this is trends over time. So it's all years across the bottom there. You can see that it isn't really straight. It looks slightly messy, but there are some cyclical trends that you can see. So a lot of what we see in large oligotrophics on these nutrient poor lakes like Christina Lake, like Slocan Lake next door, like Okanagan Lake, some of these large clear water nutrient poor lakes, we see influence of climate cycles. So we see a lot of influence from the El Nino cycle, these natural cycles, roughly on a decadal scale oscillations. So you get years of increased runoff, and we're seeing it in increased phosphorus levels that we can measure directly in the lake. So those last few dots that are clustered are, we've had quite dry years over the past 3 years, so those values have been relatively low, and in those preceding years they're higher because there's been more runoff. And again, it's not a perfect match. Sometimes there's a bit of a delay, so you can have like a really wet year 1 year, and then you don't notice that signal until the following year, and then same thing with those dry cycles. So we are capturing some of this kind of up and down natural cycles. Total nitrogen is not a limiting nutrient, but it's a useful indicator of water quality. It's kind of one of the other key ones that we measure. So sources there primarily in lakes like this, a lot of the nitrogen comes from atmospheric deposition. There's also septic leachate, agriculture, fertilizers, etc. So again, similar to the previous graph there, again, north on the top, south on the bottom. But everything here, the key takeaway point is that everything is kind of below that line. So that water quality objective, everything below that line. So as far as nitrogen is concerned, it's relatively low, and in comparison to other similar lakes, it's actually lower than some other sites there. So that's not something to worry about, but it's something that we like looking at to see if there's an increase from runoff. In some areas, like if we were to have a whole bunch of sites along the shoreline, we might be capturing some runoff in kind of like little enclosed bays or other little smaller areas. You can see that, but we have our sites out in the middle, which are overall water quality samples. Okay, chlorophyll A. So a measure of productivity and photosynthetic pigment in algae. So those are my nice little algae photos there. And again, north on top, south on the bottom there. And it follows a similar, like if I was to put these graphs on top of that total phosphorus one, it would match it actually quite well. So in years of higher runoff, you get increased nutrients and you get increased growth in algae. If you look at this seasonally, which I don't have, I don't think I do. No, I don't think I do. But over the season, if you take like bi-weekly or monthly samples, you can see when the algae levels are really quite high in the spring. So as that water mixes after the ice is off, you're getting a lot of nutrients kind of coming up from the bottom, mixing at the top, happy algae up top, and then increase in food source of the zooplankton. So you get an increase in zooplankton. And then we see a decrease several weeks after that, where the zooplankton are eating those algae, and then you don't have enough food source for those zooplankton, and then the fish are happy because they're eating those zooplankton. Again, it's just everything linked. It's an up and down cycling between these different trophic levels. Water clarity. So that's my picture that I show people. Look, this is a little check mark. It's good, nice and clear water. This is how we want things. The red X there is a green pea soup. So essentially, Secchi depth is measured with that Secchi disc. So that black and white 10 centimeter disc with the markings on that, you slowly lower it into the water, and then once it disappears, you kind of pull it back up. You kind of let it go back down, and you find that level where it just disappears, and that's what's considered the Secchi depth. And it's a really simple but effective measure of water clarity. So on these graphs here, the top is at 0, so you can think of that as the water surface, and then the little Secchi depth kind of getting dropped down. And this is the seasonal data that was collected this year from the Stewardship Society, and then the earliest one and the late one were collected by myself. So you can see how that changes, and as I was discussing with the algae. So when you have more algae in the spring, you can, there's a couple missing there because May should be the worst one. Anyways, but there's this natural cycling kind of going on. And then the next one, you can see the kind of long-term trend. So even though it's in a relatively thin band, there's a lot of year-to-year variability, but things go in relatively good patterns. So if I was to show you data from the worst example is that green lake photo there, which is Wood Lake. Everybody knows Wood Lake, which is just north of the Kelowna Airport. It's got a ton of nutrients, really good fish. But then over the past decade and a half that I've been studying it, you get to a point where the lake just can't handle it anymore, and it kind of reaches a tipping point. And that's what we're trying to avoid in these lakes like Christina Lake. So you have a lake that has low nutrients. We don't want it to get higher. There are these natural cycles that we're trying to understand, but everything on the landscape is all kind of connected and it affects it. So essentially, no algal blooms here would be great for everyone. Okay, so this here is a vertical profile. So this is just from the surface at 0 meters, going down. So temperature, you can see kind of that straight line, nice and warm, and then it sharply gets cool at that thermocline, and then it kind of gradually goes down into that colder water. So for the people who are fisher, I would say you want to go where there's the most oxygen. So you want to be fishing between 10 and 15 meters. That's the key spot that time of year in this year. So if you have one of these $15,000 to $20,000 pieces of equipment, you can know exactly where to fish, right? Next. So this was kind of how it used to be. So I just threw this up just as kind of a messy graph, but it's just the different profiles throughout the season. So essentially that red one is in the spring where things just starting to warm up, and then as it warms up, the top waters just get warmer, but the bottom waters still stay cool. But now we're trying to do these heat maps. I'm hopeful that this is a better representation of the data. Maybe it's a bit more confusing, but I kind of like it. So these are examples from a couple different lakes. But essentially in the spring, you've got that kind of blue color, which is correlates to that coldest water. And then as the season progresses, you can see the warmer water in those surface layers. And then in the fall, things mix again. So it shows you the full season, kind of cold to the warmer layer at the top, but it's always cold at the bottom. And the bottom one here is a shallower lake. So you can see the difference between a deeper and a shallower one. So the Christina Lake data is good, but it could just be extended a bit more. So this only captures the late spring, early summer through till I guess toward the end of the summer. So it would be nice to have a bit more to the bottom and extending on either side so we can see those temperature cycles. But you can see the top waters there, it gets pretty darn warm here. So here and Asayas Lake are probably two of the warmest surface waters that I've measured, except maybe during the heat dome on some shallow lakes a few years back. But generally speaking, it's some of the warmest water that I've experienced in July and especially August. This is a similar type of graph, but this is for dissolved oxygen. So for shallower lakes that have poor oxygen, so you have a lot of algae, that algae dies, it sinks to the bottom, it gets consumed by bacteria. That bacteria need that oxygen, so it's consuming that oxygen. Here, we don't really see that. We see a little bit at the north end there, where the oxygen does decrease a bit, but it's not in a really bad state. So you'll see the surface waters, just because it's warmer, it can't hold as much, so it physically can't hold as much oxygen. And then you get down to that kind of darker blue layer, where the water physics can actually hold more oxygen in it. And again, those kind of key fish layers there. And then below that, in the South Basin, is totally fine. The most bottom water, kind of 45 to 50 meters. It's a little bit low, but it's the same trend we've seen for the past several decades. So it's nothing to be concerned about, but it's just something that you won't find fish down there. Okay, so what's next? So continued monitoring at Christina Lake is vitally important to understand current conditions and how things change over time. So several years ago, we were directed by, let's just say, folks higher than me to try and just save money. And I was showing them a lot of these long-term data sets that we have. And Christina Lake was one really good example where we got long-term data. And I blocked out like every fifth data point just to show that, well, you can't really see any of those trends. And then I kind of removed a couple and then, oh yeah, I can see some trends. And then you remove a couple more. And it's not until you fully realize that you need this annual sampling. And I'm doing a bare minimum. I'm coming here twice a year to do this water quality sampling. It's a bare minimum, but it's a good way to understand water quality over the long term. And most people don't study water quality over the long term. They're just little snapshots in time sporadically. And we're really trying to just get more. So I managed to convince people that it's worth the money, it's worth the investment to do a long-term trend sampling. So we will continue this work early spring and fall as part of the network. So it's not a one-off project, it's all connected. So this is just one part of a larger provincial monitoring network. And Christina Lake Stewardship Society hopefully will continue the seasonal second depth and temperature and geo profiles. However, a couple of points here. So I do suggest if you can sample, just increase it on the outer bounds, so more spring and a little bit more fall if possible, and go right to the bottom if you can. And next, if I have a minute and a half, maybe I can just show you a couple of updates. And so over the past several years, we've been working on some new websites and monitoring tools. So there's a website. Easiest way is to go into Google and just type BC Lake Monitoring, and this will be the first one. We worked hard to get the first one, BC Lake Monitoring. If you can't remember the actual URL there. Next. So on that little handy dandy map, interactive map there, brings you to the map portal, and this is an interactive one. So you'd have to kind of go down to where Christina Lake is. You can click on a dot, a little menu pops up, and it'll have a whole bunch of metadata. So it'll have information about the lake site, the depths, and then there'll be links to data reports. So all of the data that we've been collecting are now housed in there. And I don't know about you, but I'm a visual kind of person, so I like maps, and I find them very handy. And there's a lot of great information in there. So I'm just barely scratching the surface. So if you want to geek out about water quality, about lakes, I could talk about it whole day kind of thing, but I won't. So I will direct you to the map portal. The other thing I want to highlight, which wasn't in existence when I was here last in 2019, is Algae Watch. So this is a new initiative we have. So essentially, when people see algae blooms on lakes, especially if they're homeowners with water intakes, they're very concerned about this sort of thing. So we have an education portal here to, first to educate people. We collaborate with folks because I don't live at these sites. I don't know fully what's going on. So this is kind of a reporting site as well. So you can go on there, we can track it, and we can better communicate things. So these things that I'm showing here, so that first one is a bad algae bloom. The next one might look bad, but it's actually beneficial algae. You might see this kind of brown iron staining with this, which kind of might look like in oil or gasoline spill, but it's most likely this iron oxidizing bacteria. Everybody safe. And yeah, so and the last one I showed up just because it's the most striking, which is purple sulfur bacteria. You will not come across this except for a few very rare and interesting lakes in the province. So remember what I was saying about lake mixing? That comes from a lake that never mixes and hasn't mixed for probably over 10,000 years. So you have these super distinct layers, doesn't mix, and then you get these really unique communities of bacteria living in there. So you won't come across it, but I just thought it's super cool, so I thought I'd share it. So on this website, we've got a ton of information. So essentially, if you see things that look like potential algae bloom, I recommend going to the website. It might not be algae, it might be pollen. We've had calls from here about people saying this, they're kind of like yellowy scum on the surface, a lot of pollen or just different natural phenomena. We've got a ton of photos in there for natural phenomenon. But if there are algal blooms ever, then this is the place to go. And it's not just for here. Any water body you go to, you can check out this site. Next, that's just the map. Next, and that's it. All right, thank you very much.
Got time for one or two quick questions. Yeah?
I don't have a question for you, but I'll give you a little quick information. You know how deep Christina Lake is?
The deep spot that we have was 51 meters.
Oh, meters, okay, no, I have to convert to feet. 187 feet. I've been down to 135. I didn't get to 187.
Okay.
Yeah.
Yeah. So
diving, like
the deep spot, according to the old bathymetric maps, is almost kind of due northwest of Texas Creek Boat Launch.
Yeah.
Yeah, not too far. Yeah, right across from there, right in the middle.
Been there many times.
Yeah.
Okay. Are we good, everybody? Thank you, Mike.
Okay, thank you. Thank you very much. I do have to leave. I thank you again for everything. Thanks for inviting me, and thank you for the lovely lunch. Always come back to the lunch.
Priority.
Okay, next up is our very own